Perfect. So, hi everyone, and welcome to day two of our two-day trainee research presentation series, New Perspectives on Menstrual Health. For those who joined us at our first session, it's great to have you back. And for those who are new, we're really glad that you're here. I'd like to start today's event with a land acknowledgement. We recognize that we live, work, play, and participate in community on the unceded, ceded, and traditional territories of the 630 First Nations, each of which possess their own unique traditions and history here on this land that we now refer to as Canada. We acknowledge the importance of the Truth and Reconciliation Commission of Canada's call to action and the United Nations Declaration on the Rights of Indigenous Peoples. In all of our work, we are committed to ensuring Indigenous women's rights to health and safety and the equal opportunity to participate in a manner that recognizes and respects Indigenous cultures and traditions. Today, I am joining you from Toronto, which is covered by the Toronto Purchase Treaty Number 13 with the Mississaugas of the Credit. Toronto is now home to a vast diversity of First Nations, Inuit, and Métis who enrich the city. Many of our members live in Vancouver, which is part of the unceded homelands of the Coast Salish peoples and the traditional territories of the Musqueam, Squamish, and Tsleil-Waututh First Nations. In our previous event, we began to explore how complex and often overlooked menstrual health is, and today we're continuing that conversation. Menstrual health is often treated as something routine, but people's experiences are anything but. They vary widely, and they are not always well understood or well supported. Too often, symptoms are minimized, and people are left without clear answers or care that reflects what they're actually experiencing. And creating space for this work really matters. It allows us to build a more complete understanding and to move toward research and care that takes menstrual health seriously. So thank you to our trainees and to all of you for being here. So first, today we have Carla Maduda speaking, and I'll just have you share your slides when you're ready.
Can everyone see my slides? Okay, perfect. So hi everyone, I'm really happy to be joining everyone today. I'd like to thank the organizers for inviting me to speak. My name is Carla Maduda. I'm an MD PhD candidate at the University of Western Ontario, which is in London, Canada. And I'm in Dr. John McCormick's lab. And today I'll be talking a little bit about my PhD work, which looks at unconventional T cells in menstrual toxic shock syndrome. I cannot switch to my next slide for some reason. Okay. Did that switch the slide?
Yeah,
Okay,
it's
sorry
good.
about that.
All good.
So, menstrual toxic shock syndrome, or menstrual TSS for short, is a life-threatening disease that occurs at menstruation, as the name suggests. And some of the symptoms include a high fever, rash, multiple organ dysfunction syndrome, which leads to organ failure, and ultimately death if left untreated. So, menstrual TSS is a very rare condition, but it is very serious. And there's approximately one case in every 100,000 people who menstruate. There's often a misconception that menstrual TSS is caused by the use of high absorbency tampons. In reality, there is an association with the use of tampons, but it's actually caused by a bacterium called Staphylococcus aureus. Staphylococcus aureus, or Staph aureus for short, is a common bacterium that's found on the skin, in the nose, and in the vagina. And up to a third of the population is colonized with Staph aureus. So what this means is that Staph aureus is persistently found in these different areas, and more often than not, it doesn't actually cause any harm. So it's pretty harmless, but under the right conditions, it can become pathogenic. So some of these illnesses include pneumonia, which occurs in the lungs, infective endocarditis, which occurs in the heart, sepsis, which is when the bacteria enters the bloodstream, osteomyelitis, an infection in the bones, both menstrual and non-menstrual forms of toxic shock syndrome, as well as skin and soft tissue infections. So, the human immune system is composed of dozens of different white blood cells, and their primary function is to kind of ward off different types of infections. So, these can be bacterial, they can be viral, they can be fungal. And essentially, they're supposed to be able to clear infections and make sure that the human body responds appropriately. However, Staph aureus can kind of manipulate the typical immune response through the production of something called a superantigen. And before I get into what a superantigen is, we must first understand how a conventional immune response works. So, of the dozens of different white blood cells or immune cells that we have, there are two that I'll be focusing on. So, we have an antigen presenting cell, which consistently samples the environment and looks for molecules just in the environment, and it looks specifically for pathogenic ones. So, for example, those that come from bacteria or viruses. Then you have a T cell, which can recognize these pathogenic molecules. So you have this interaction between these two receptors on the surface, and the interaction is extremely specific. So it relies on this pathogenic molecule that's being presented. So much so that only about 0.01% of all T cells become activated. So it's a very, very extremely specific interaction. When you have a staphylococcal superantigen present, you actually get bridging of these two receptors in a fashion where the pathogenic molecule that's being presented no longer matters. So in this situation, you now have about 20% of T cells becoming activated, which is a 2,000-fold increase in the number of T cells that become activated or turned on. So this puts the immune system into overdrive and can result in toxic shock syndrome. So, Staph aureus can produce 26 distinct superantigens, but what's really strange is that only one of these 26 can cause the menstrual form of toxic shock syndrome, while the others cause non-menstrual forms. And interestingly, this one superantigen is also responsible for about 50% of the non-menstrual cases. So there's something strange going on here, but we don't know what or why. What I didn't tell you about the molecule that's on the T cell surface is that it's composed of something called a V beta chain. So there's dozens and dozens of these different types of V beta chains within this receptor. But each superantigen can target kind of specific ones. But the menstrual TSS superantigen only targets V beta 2. The other 25 staphylococcal superantigens cannot target this molecule. So this kind of leads to the rationale for my work, which is that there's a population of unconventional T cells called mucosal associated invariant T cells, or MATE cells for short, and they have significantly more V beta 2 molecules than conventional T cells. And this has been shown in the literature as well as in some of my work, which in the figure on the right here, I'm showing two different types of T cell populations on the x-axis, as well as the percent of V beta 2 positive molecules on the y. And conventional T cells, about 7.5% of them have this V beta 2 molecule, while when we look at MATE cells, about 15% of them have this molecule. So this leads to my hypothesis, which is that MATE cells are more responsive to this menstrual TSS superantigen and are therefore significant contributors to the immune response. To investigate this, in my work, we recruit healthy blood donors in our lab. We draw their blood and isolate the white blood cells or the immune cells, and from there we expose them to the menstrual TSS superantigen. We let them incubate for about 24 hours, and then the readout here is flow cytometry. So this is a method that allows me to sort out different populations of cells. So I can look at conventional T cells, I can look at MATE cells, I can look at active cells or non-active cells, as well as cells that have this V beta 2 receptor or not. So in my first experiment, I wanted to see how do conventional T cells compare to MATE cells in terms of the response and activation, or the ability to turn on when exposed to this menstrual TSS superantigen. So I went through my workflow, I isolated these immune cells, and then I looked at three markers for activation. These are CD69, interferon gamma, and TNF alpha. It's not important to really understand how each of these function, rather appreciate that they're signifiers that these cells are becoming turned on. What we see with CD69, which is this first graph on the left, we see that MATE cells express significantly more CD69 in comparison to conventional T cells when they're exposed to this menstrual TSS superantigen. We also see an increase, although not significant, with interferon gamma, which is this panel in the middle. And finally, on the right, we see significantly more TNF alpha expression from mate cells than conventional T cells. So this signifies that they're significantly more activated relative to conventional cells. In my next experiment, I wanted to introduce actual bacteria. So to do this, I used a clinical isolate from a menstrual TSS patient, and we call this our wild type strain. So we've done nothing to it. We've just essentially isolated it from a patient. And on the right, we used that same strain, but we've genetically modified it so that it can no longer produce the menstrual TSS superantigen. So essentially, the two strains are identical with the exception of the ability to produce that superantigen. I then grew the bacteria overnight, removed the bacteria, and collected their metabolites, which is this kind of liquid that remains. So these metabolites that are left over should contain superantigens. From there, I exposed immune cells to these leftover metabolites and then performed the same types of experiments again. Here, I narrowed in specifically on mate cells that express the V beta 2 receptor, which is again the target for the menstrual TSS superantigen. I once again looked at the same three activation markers, and what I found was that the strain that we consider kind of our wild type resulted in a very high amount of CD69 expression from those V beta 2 mate cells. But the strain that no longer produced the menstrual TSS superantigen, we see a significant reduction in CD69 expression. Again, with interferon gamma, which is in the middle panel, we see a reduction, although not significant. And finally, on the right-hand side with TNF alpha, we see a significant reduction in expression with the absence of the menstrual TSS superantigen. So what this indicates is that these cells are becoming turned on in the presence of this superantigen, and it's not some other type of metabolite that the bacteria are producing that's turning on these cells. And finally, the last piece of data I'll be showing you today is what happens when we actually remove the mate cells prior to exposure. So to do this, I call my kind of conventional workflow the complete cells. So I just go through my workflow as normal and isolate immune cells. In my mate deplete cells, I add an extra step where I use magnetic beads to draw out the mate cells. From there, I use these two cell populations and expose them to the menstrual TSS superantigen and see what happens. And the thinking here is that by removing the mate cells, we should see a reduction in activation. So once again, looking at those same three activation markers, I narrowed in on the V beta 2 cells, which once again is what that menstrual TSS superantigen targets. And we see that in all three instances, when the mate cells are removed prior to exposure, we see a large, if not significant, decrease in activation of these cells, which signifies that they're major contributors to the activation and inflammatory response. To wrap everything up, mate cells are this unconventional T cell population that respond to the menstrual TSS superantigen more profoundly than conventional T cells do. And this is likely due to their high abundance of the target for the menstrual TSS superantigen, V beta 2. And finally, this work contributes to our understanding of menstrual immunity and safety. With that, I'd like to thank my lab, all the blood donors who made this work possible, as well as our funding sources, and I can now take any questions.
Thank you so much, Carla. So if anybody has any questions, you can leave them in the chat or you can ask them yourself as well by unmuting. We have 1 minute for a quick question. If not, I'm just curious. Can you tell us a little bit more about where the idea that using high absorbency tampons might increase the risk for toxic syndrome?
Yeah, so although it's not the focus of my PhD work, I did a lot of work on this during my undergrad and in my master's degree. And
Hmm.
essentially, it boils down to changes in the environment that occur at menstruation. So generally, the vagina is a very acidic environment with a pH ranging from about 3.0 to 4.5 under non-menstrual conditions. However, at menstruation, we have this large drop in estrogen that occurs, which results in a rise in the pH level to about near neutral. So this is one of the factors that changes. The drop in estrogen also results in a large decrease in glucose levels. And what we know about glucose is that it actually stops Staph aureus from producing the menstrual TSS superantigen. So under normal
Oh.
conditions, or I guess not normal, but non-menstrual conditions, when
Mhm.
there's high glucose levels, the bacterium can't produce the superantigen. But at menstruation, when the glucose levels are much lower, the superantigen can be produced, and this is when you can have menstrual TSS occur.
Oh, super interesting. Thanks for sharing. So I'll just have you switch your slides over. And if anybody has a quick question, you can also drop it in the chat and Carla can comment on the chat. So next we'll have Jaza Alam, if you can share your slides.
Is it visible?
Yep, looks great.
Okay. Hello everyone. I am Jaza Al. My talk, my talk's topic today is genetic risk pathways in endometriosis and its early diagnostic potential. This work was done when I was an undergrad at University of Toronto, in collaboration with the research team at DNA Labs. So, endometriosis is a chronic condition where tissue similar to the lining of the uterus grows outside of it, often on the ovaries, fallopian tubes, or surrounding pelvic structures. It affects about 1 in 10 individuals of reproductive age, making it one of the most common gynecological conditions. But despite this, it takes almost a decade to diagnose. And even when there is a diagnosis, it requires an invasive surgical procedure, which can only detect it when the disease has progressed significantly and a lot of damage has already been done. So this creates a significant gap between the disease, when the disease begins and when it's actually identified. So you must be thinking, why does it take so long to diagnose endometriosis? When we look at the symptoms, one of the key challenges is that they are highly variable between individuals. Many of them experience chronic pelvic pain, painful periods, and in some cases, infertility. And these symptoms are also frequently dismissed or normalized, which further contributes to the delay. Clinically speaking, these symptoms are non-specific and overlap with many other conditions, which also makes it harder to kind of identify a clear clinical pattern early on. However, this variability is not random. It actually reflects the underlying biological complexity of the disease, and this is where the challenge begins. So endometriosis is driven by multiple systems, including inflammation, oxidative stress, and estrogen dysregulation, all kind of interacting with each other. And as a result, diagnosis is delayed until the symptoms are very severe. So if endometriosis is driven by these underlying biological processes, an important question becomes, can we detect these changes earlier before the symptoms become severe? And rather than waiting for clinical symptoms or surgical confirmation, could we identify measurable signals within the body that indicate increased risk? And this is the central idea behind my research to explore whether underlying biological patterns can help us move toward earlier detection of endometriosis. So, to explore earlier detection, we first need to establish whether genetics plays a role in endometriosis. And there is a large and growing body of research, including hundreds of genetic studies and genome-wide analyses. supporting a significant genetic component. Also, as we can see that individuals with a first degree relative are affected, like have a 5 to 7 times higher risk of getting affected. And in this context, we focus on SNPs or single nucleotide polymorphisms, which are these small genetic variations that can influence how key biological pathways function. It is actually one of the most common types of genetic mutations, evolutionarily stable as well. And this allows us to examine underlying biological risk rather than relying only on symptoms. So in terms of our research approach, we used a cross-sectional genetic analysis based on data from the FERN test, which is a clinical nutrigenomic panel, and it evaluates kind of fertility-related genetic variants and multiple genes related to inflammation, oxidative stress, estrogen metabolism, and reproductive function. So for each variant, we compared the allele frequencies in our cohort to kind of global reference populations to determine whether any risk variants were overrepresented. And then we grouped the significantly enriched variants based on their biological pathways to better understand endometriosis. So here are some of the key findings. On the left, we first observed that several of the genetic variants showed increased frequency in our study population compared to global reference data sets. And these included variants such as IL-6, CRP, SOT2, which are all previously implicated in reproductive and inflammatory biology. And what was particularly notable was that these variants, instead of being distributed randomly, they were actually clustering around biological pathways, which were all related to inflammation, oxidative stress, hormone metabolism, and endometrial function, which are all involved in endometriosis. So further on, when we mapped the genetic variants to their biological function, and we observed clear clustering into key pathways relevant to endometriosis. So, for example, variants in IL-6 in CRP are involved in inflammatory signaling and immune activation, and variants in SOT2 and GSTM1 relate to oxidative stress and cellular damage. Similarly, MTHFR and PMT influence estrogen metabolism and methylation processes, while VDR is linked to immune tolerance and endometrial receptivity. So together, these variants sort of converge on biological systems that are central to endometriosis pathophysiology. These findings kind of suggest that endometriosis is not entirely invisible at the biological level, like how it's completely unknown when symptoms become severe. Instead, there are measurable genetic and pathway-based signals that reflect the underlying risk. So this allows us to identify risk earlier than diagnosis and move toward non-invasive screening. So moving forward, the next step for us is to validate these findings in a larger cohort of individuals with confirmed endometriosis. And we also aim to include women with unexplained infertility, given the known overlap with undiagnosed or silent endometriosis, which is when symptoms are not very overt. And you find out when it's too late, after like a decade or two. And we also want to focus on gene-gene and gene-environment interactions to better understand the variability in the disease. And ultimately, we want to use all of that analysis and data to kind of develop polygenic risk scores that could be used for early risk stratification. And this would potentially integrate genetic, clinical, as well as lifestyle data to support more personalized reproductive care. So, to conclude, improving outcomes in endometriosis would require continued awareness, research, and importantly, listening to patient experiences. And the goal of this work is to add another layer of information that could support physicians in earlier and more informed decision making. So ultimately, our hope is that genetic risk profiling and pathway-based insights can contribute to earlier recognition and better care for patients with endometriosis. Lastly, I'd like to thank my DNA labs and Fern DNA testing team, under whom I carried out this project. Uh, these are my references. That's all. Thank you. Sorry, I'm not able to hear.
Oh,
Yeah.
I think I was speaking, but I was muted, of course. I just said I love the cartoon.
Thank you.
Okay, so if anybody has any questions, again, feel free. And I think your messaging towards the end there really resonated with a couple of people in the call on the call today. I saw a couple of reactions. I'm not sure if you saw them while you were presenting, so just wanted to pass that along in case you didn't.
Okay. Thank you so much.
I was actually thinking while you were presenting about your work, what kind of implications there are and what you think would be a really cool next step. And I'm interested that you mentioned a polygenic risk score. And I'm sorry if I misunderstood, but did you mention also incorporating lifestyle factors into that?
Yeah, definitely. So for polygenic risk score, I was really interested and I'm really excited about this,
Hmm.
because as we can see in a lot of kind of pilot clinical programs or medical institutes in the US and UK, they're now considering it or making it a norm to involve polygenic risk score analyses. And for lifestyle, it kind of goes into the field of sort of nutrigenomics, where some people are more compatible with certain types of drugs or more receptive, and some people also would have higher deficiency of iron or other sort of supplements or so we kind of want to include that as well, and see if all three kind of factors, clinical, genomic, and lifestyle factors, can kind of help people manage this disease. Because in a lot of cases, in the beginning, it's like it's dismissed, and usually the solution is just to have a bunch of painkillers, which are also kind of bad for you. So yeah, that is the idea of it.
Oh, somewhat unmuted, but maybe it was just an accident. Yeah, that's fascinating. I think it's great that you're really thinking towards the direction of integrating multiple perspectives because I was going to ask you a little bit more about the limitations and ways to kind of complement genomic focused studies. So that perfectly addresses that question. Thank you. Okay, if there aren't any other more questions, we can switch over the slides to the third speaker. And again, feel free to leave comments in the chat if we've moved on. Okay, so our third speaker today is Alexandra Kane, and we'll have you get started whenever you're ready.
Okay, let me know when you guys can see it.
There, we see it. It's beautiful.
Okay, perfect. Thank you so much. So, hi everybody. My name is Alexandra Kane. I'm a graduate student in the Masters of Interdisciplinary Health Sciences program at the University of Ottawa, and I'm working with Dr. Angel Foster as my supervisor. Today, I'm going to talk to you about my thesis research, which is exploring menstrual hygiene management in acute humanitarian settings, and it is a multi-method study. Okay. So this is just an agenda to give you a rundown of the topics I'll cover. So we'll start with the intro to give a bit of background information on the topic. We'll go to my rationale, the research questions which are guiding the research, the methodology that we're using, some of the expected results and the preliminary results, because We're still in the middle of doing it. Some of the limitations that we've identified, the significance of the findings and the study, and we'll do a bit of positionality and reflexivity. So I'll kind of share my perspective through this whole thing. So to give you a background information, we'll start off, I'll first kind of start with menstrual hygiene management. So menstrual hygiene management refers to the comprehensive management of menstruation through access to clean, absorbent, and culturally appropriate menstrual materials, as well as private facilities for changing, bathing, and disposing of used materials. That is the UNICEF definition. Now, a lack of access to menstrual hygiene management is a very widespread issue that targets many populations around the world, majorly in low and middle income countries and especially in crisis settings. This is called often referred to as period poverty. So you might have heard of period poverty in the news before. Um, the downsides of period poverty, some of the health impacts that we have seen are a much higher risk of urinary tract infection, reproductive tract infections, in maybe cultures where there's some stigmatization of menstruation. There's experiences of anxiety, psychological distress, and as well, it also increases the risk of gender-based violence. Now we'll set up the other half of the study, which is the humanitarian crisis setting. So the acute phase of the humanitarian crisis refers to the period directly succeeding the disaster. So after the onset of a disaster occurring, which is when normal life of a community is disturbed. It's usually characterized by major human life loss, illness, and or widespread displacement. By the end of 2026, an estimated 136 million people worldwide will be forcibly displaced. So that's actually almost, I think, two times more of the estimation. Essentially, like by the end of 2024, there was 122 million displaced. So this is already a huge increase by the end of 2026. So we know that 51% of this population are women and girls of reproductive age. This is a major population that needs to be addressed, and their needs need to be addressed in a crisis setting. So during the acute phase, usually relief and response efforts are focused on life-saving aid. And so that might be provisions such as potable water, emergency shelter, life-saving medicines, electricity, food, these kind of things which are considered life-saving. Now, the way that the humanitarian architecture, which is sort of the big collection of all of these agencies, how they work together, there's this thing called the Intersectoral Standing Committee. And this is a committee of agencies and organizations that work together, and they're each focused on a certain cluster of response at a time. These clusters will shift every few years. So at the moment, I mean, generally health will be covered by the World Health Organization, but amongst some of the other organizations, there might be some shuffling. But the idea is that they all work together to coordinate response during crisis settings. Complex issues that can span several areas of concern. So if this isn't something as clear-cut as, health covered by World Health Organization, if there's more complex issues that don't fit cleanly into one cluster, they will often be considered intersectoral, and they might become something called a subcluster or an area of responsibility. And essentially, this is a deliberate coordination between several clusters in order to cover this sort of complex issue. Now, a great example that we have that exists is actually the area of responsibility. It's the subcluster of gender-based violence. So I mentioned that earlier. This is actually something that can be connected to menstrual hygiene as well. Now, gender-based violence is different types of harm that are perpetrated based on socially ascribed gender-based power inequalities. And we have seen over the years, essentially, it was identified that gender-based violence as a topic in a crisis or a disaster setting is sort of controlled or influenced by several different, I guess, clusters at the same time. And therefore, there needs to be there needs to be a coordination between clusters in order to mitigate harm to these populations. So this includes some action through collaboration between the food security cluster, the water sanitation and hygiene cluster, the camping coordination management cluster, and the interagency standing committee, which again is sort of like this group here that runs everything together. They actually established guidelines, a set of guidelines, standardized guidelines, which dictates the protocol and the procedures surrounding gender-based violence mitigation. That's what this QR code is here. It's actually a link to these specific guidelines for gender-based violence. And this is relevant. I'll circle back to this. The reason this is relevant, and I use the example of gender-based violence, is that while that is an intersectoral and complex issue that has standardized guidelines to control its procedures, menstrual hygiene management is in the same way an intersectoral issue, but it is not coordinated as an official subcluster or area of responsibility. So this is a complex topic. We know it's a complex topic. It needs to be managed by a few different areas of the humanitarian architecture, and yet there's not really any standardized guidelines that's enforcing that at the moment. We know we see through previous research, we know that gender bias and stigmatization of menstruation can lead to the deprioritization of MHM in policymaking and humanitarian response. We also know that menstrual products are frequently underfunded or omitted from emergency aid packages. Another sort of thing that some researchers have identified is that potentially there are some discrepancies between the definitions of MHM. So one organization might consider MHM clean and absorbent materials, while another considers MHM clean absorbent materials and water and private spaces. Another might include education surrounding menstruation as a component of MHM. So there's some discrepancies that have popped up, and I'll circle back to that again once we get to my preliminary findings, but that's sort of a theme that seems to be occurring is there's a lot of discrepancies within the architecture. This brings me to my rationale, which is that gaps clearly exist within the global access to MHM, specifically in the acute phase of a humanitarian crisis. And through my thesis, I'm hoping to sort of highlight, elucidate some of these specific barriers that exist within the policy and practice standards. My research questions. My main research question is, what do we know about MHM initiatives in acute humanitarian settings? What are the facilitators, barriers, and best practices related to MHM initiatives in acute humanitarian settings from the perspective of key informants? And finally, how can MHM initiatives in acute settings be improved? Start with the scoping review. So currently I'm at the sort of extraction and charting phase of the scoping review. I'm using the ARCSI and O'Malley framework as adapted by Levac and colleagues. And here is my Prisma chart. So you can see we were able to pull out 2,632 relevant articles, but it has been whittled down to 38, which will be included in the review. What are we going to be looking for and charting within our scoping review findings? So we are looking for details about the publications. Does it focus on the perspective and experiences of crisis-affected populations, or are these publications focusing on the intervention itself and the response protocol itself? We'll also be identifying the setting of these articles. So where specifically geographically are most of these articles focusing on, as well as these sort of temporal settings. So are these studies focusing on the acute phase of the disaster or the protracted, which is more the long-term phase of a disaster response? These are the scientific databases that we are using or that we used rather, to find our articles. We used Scopus, Medline, Global Health, CINAHL, and we'll also be doing a gray literature search as well. For the key informant interviews, so this is going to be about 20 to 30 key informants, and we are hoping to recruit representatives from NGOs, some researchers, people that work in public and private relief agencies, and they will be identified through purposive sampling, through publicly available information, and also we'll be using snowball sampling. So of course, if we identify one eligible participant, tell them to use their networks and seek out any other relevant participants. Data collection. So each structured interview, or rather semi-structured interview, will be about 1 hour, and they'll be virtual, but there might be some in person if applicable. Prior to each interview, we will collect the oral consent of each key informant. They will be recorded and transcribed later, and I will also be taking notes during the interview and writing analytic memos after each interview to make sure I'm keeping everything in check. For the data analysis, I'll be using the Ritchie and Spencer applied framework analysis as outlined by Gail and colleagues. And I won't list off every step here, but we have the steps here which will be used for the analysis stage. So, like I said, transcription, listening, noting observations, extraction, and it keeps going. Once I finish both the scoping and the key informant interviews, I will be doing some component integration to try and identify if there's areas where there's a lot of concordance or discordance between the two components. And I'm going to essentially try and fill in the gaps between those two segments and see where there's some connection or discordance between them. So here are some of the preliminary and expected results. So because we're mainly through the scoping reviews, some of the results that are starting to pop out quite a bit is that within the research that we're assessing here, there is quite an absence of research that focuses on the acute phase, which is really what we're looking at. So me and my co-reviewer, I believe we were using essentially around 9 months in terms of like timing. So about 9 months post-disaster is what we would consider the acute phase of the disaster. And what we're seeing is that a lot of these articles and, or rather, a lot of these studies that have been conducted are focusing on a protracted refugee camp, which might, they might be interviewing participants 2 or 3 years post-disaster. So that's really more of a long-term focus and that's popping up quite a bit. We are really seeing if there's any acute focus, like right at the onset. We're also saying, as I sort of mentioned earlier, that there is a bit of a discordance between the definitions. So some articles that are assessing experiences of humanitarian aid workers have reported feeling confused because they're not quite sure, well, what is considered MHM? Should I be doing this? Should I be doing this? There's a bit of discordance there, which is causing some gaps. The KI interviews, so the key informant interviews, are expected to be done by the end of this summer, and we're hoping they'll give some insight to the staff training concerning MHM, which might answer that definition question, and how exactly are the directives that they receive, how are they translating into practice on the ground level? Once I complete my thesis, I intend to submit the articles for peer-reviewed publication. Some of the limitations, because we're using purposive sampling, there is a possibility that there could be scheduling conflict and there's sort of some hitches that might pop up in there. So I will be really trying to recruit as many participants as possible to mitigate if there's any dropouts. And there's also a bit of a constrained timeline. I'm doing this in the context of a master's thesis work, so I don't necessarily have years and years to do it, but I'm really hoping to attend as many conferences, events, network as much as I can, make sure I can recruit as many participants as possible, and reach my goal. So the significance, which is that if you turn on the news, you can see there are many conflicts going around the world. Climate change means that there is quite a bit of, ecological disasters occurring. Displacement will continue to occur, acute crises will continue to occur, and so the health of menstruating individuals really has to be prioritized in humanitarian relief protocol and efforts. And I'm hoping to highlight some of the practices and gaps, and also the facilitators, if they exist, to operationalizing MHM initiatives for the future. Quickly, I'll just go through my positionality. So I am a white, cisgender, able-bodied woman of reproductive age. I was born and raised in Canada, and I've always had consistent access to clean water and menstrual hygiene materials. So I have a basic education on the humanitarian sort of international relief efforts, but I've never personally contributed in a volunteer or professional capacity. I aim to approach my key informant interviews with a receptive mindset that is conducive to documenting their perspectives. And throughout this whole process, I will be engaging in a formal reflexive process, including I'll be doing some memoing debriefing after each interview to understand the ways in which my positionalities might influence the researcher-participant interaction. Thank you so much. And if anybody has any questions, thank you for listening to me.
Thank you so much. We do have a question already in the chat. Emily, did you
Sure.
want to ask it yourself, or I can read it out for you?
Oh yeah, sure. Thanks for an awesome talk, Alexandra, and all the speakers actually. I was just curious how you're defining disaster in your scoping review because I can imagine that there might be differences in the literature with humanitarian access if you're talking about like hurricane, monsoon, like natural ecological disasters versus war or pollution-related displacement, like human-induced disasters. So I'm just curious
Yeah.
how you're approaching that.
So for the scoping, our search strategy was to cover human-made and ecological as two sort of branches, but they're both being included. So some of the searches that I've sort of organized them, right? I have like chunk one, which is like related to menstruation. So my keywords are menstrual hygiene, sanitary, sanitary products, sanitary, feminine products. So all the different variations of menstruation is one chunk. My second chunk, which is humanitarian, or rather human-made disasters. So I'm using keywords like ethnic cleansing, war, armed conflict, forced displacement, political disaster, I believe is one of them. And my third chunk, which is the ecological, and that includes keywords like natural disaster, and then we go down like hurricane, tornado, typhoon. We're covering like all of the possible sort of disaster related themes, keywords, because yeah, as you said, it depends, one researcher might not include the word disaster, but they might be referring to like flooding that displaced 3,000 people, which is technically a disaster, but they don't use that keyword. So we're just we cast a very wide net to make sure we're getting all those keywords in there for the disasters.
Thank you so much, Alexandra. So we just get you to switch your slides so
Sure.
Simul can put theirs up. In the meantime, I just had a quick comment, I guess. I think it's really fantastic that you're being so thoughtful with your methodology. Sorry, Simul, if you can just share your slides in the meantime. Okay, perfect. And you did mention that you will be taking geographical location as one of the considerations, but I'm also wondering about cultural considerations. Is that
That's
worked
actually
into your... Sorry, go ahead.
it. That's okay. So because we're sort of just at that stage right now, that's actually a really great suggestion. Um, I think like for my data extraction table, I have like geographic location. I have like a setting of the publication, like so setting of the study, but I was considering actually including another code or another like column to include, which would be origin of the authors, because there is a lot of this is sort of a topic that has a lot of nuance, like cultural nuance as
Hmm.
well.
Hmm.
So it's going to be different if um, if like myself, like a white cisgendered Canadian researcher is interviewing people in, crisis affected people in Lebanon versus a Lebanese researcher, interviewing people in Lebanon. So there is a
Right.
sort of like cultural nuance. So that is going to be one of my points to be extracted in the table, which will be like, what is the origin of the author and what is the setting of the study? And we'll see if anything is sort of like emerging for that.
Yeah, wonderful. Thank you for that. We hope to hear about your findings at a later presentation date.
Yes.
Okay, so last but not least, we have Simul Masood, who will be presenting for to close out today's event. So go ahead whenever you're ready.
Hello, can you guys hear me? All right. Hi, everybody. My name is Simmal Masood, and I am going to talk about the link of PCOS in particular to the molecular blood-based biomarker patterns linking to reproductive cancer. Now, I did my research in Pakistan, so I was really interested in what Alexandra had to say with LMICs and working with in countries with a lot of disaster. So, by the way, if you ever let me know if you want some people from Pakistan. So I did my research in Forman Christian College University, where I was working on the patterns that are prevalent in. So I was looking at the biomarkers that are prevalent in reproductive cancer and PCOS. So in particular, um, one second, is it moving ahead for you guys?
Yep.
Yeah. All right. So in particular, my research was exploring oxidative stress, PCOS-driven oxidative stress, because it was thought. So there was just just like a lot of literature PCOS, combining oxidative stress to both PCOS and cancer. But I wanted to see if this bridge was between those two conditions also. So now, one big problem that I faced was that there were very few data sets that were talking about a South Asian genome, which is why this was one of the first studies to talk about PCOS in terms of South Asian genes, and also to see reproductive cancer being linked with oxidative stress. So what we did was we collected data from cancer patients who had PCOS. We collected data from PCOS healthy individuals and healthy individuals, and we ran molecular analysis that was performed via, of course, we did RNA extraction, DNA extraction, we did RT-PCR, and then later on we ran our analysis, and we were able to find significant down regulation of certain genes, for example, PTEN and NFE2 in cancer subjects, which was expected, while PCOS samples exhibited more stable expression levels, which was not expected since they also PCOS is also thought of to be due to oxidative stress. And there has been a lot of research on this, but for some reason, the expression levels were stable in most cohort. So again, like I told you, this was one of the first studies that was done across three different across reproductive cancers, in which we included vaginal cancer, um, endometrial cancer, we included uterine cancer, and I even included cervical cancer to have a better review. Now, just to talk a bit about PCOS, if somebody is unaware, PCOS is a prevalent endocrine disorder, which is now thought to be a metabolic disorder more than an endocrine disorder, characterized by an ovulatory dysfunction and hyperandrogenism. There's a lot of debate about what can be PCOS symptoms and what can't. There are a lot of diagnostic criteria that differ in some criteria, but mainly the criteria that we follow here is the Rotterdam criteria, and the single best way that doctors here determine if you have PCOS or not is through an ultrasound test. So, so oxidative stress is a major contributor to PCOS, where we see that so there is this imbalance between the reactive oxygen species and antioxidant defenses, which can lead to insulin resistance, which is a very common symptom of PCOS. Now, insulin resistance is also common in other metabolic diseases. Also, it's also common in diabetes. Again, diabetes, Pakistanis have one of the highest. They're at the highest risk for diabetes, so they already, like, most of the population already does have genes for diabetes. So I also wanted to see if those diabetic or those genes that eventually lead to diabetes are also prevalent in people with PCOS. So we did see, I actually ran my research on 8 genes. There were a few for cancer, there were a few that were for insulin receptors, and there were a few that were targeting hormonal dysregulation. So we did see that there was a critical, and so during our work, we did see molecular signatures that were pointing towards some genes. So there was the one of the genes was for oxidative stress, and one of the genes was for cancer, which is PTEN, pretty famous one, that was significantly downregulated across the study groups. Now, again, the data collection in Pakistan is a bit different because this is how we used to do it. There is this is again one of the biggest hospitals in Pakistan. So this is how the data is collected. I had to physically whisk through every single patient file, take pictures again with consent, but again, it takes 15-20 minutes to take pictures to assess, okay, whether they fulfill the criteria or not that I'm looking for. And to get the again to draw blood, even it was a huge. Even when I had the consent of patients, there were nurses who weren't letting me draw blood, or they weren't drawing the blood themselves. So there were like a lot of barriers that I had to face. This is where they were treated. This is the place where they were sitting down for their chemotherapy. So a bit bleak, to be honest, but this is a public hospital, so you would get all types of patients from all across Pakistan. You would get all you would get patients from all types of socioeconomic backgrounds. So the sample in itself was really rich because of that. We so there were like again I had to whisk through everything and I had to see if everything was correctly written. So data collection in itself was very, very manual. And also to know which person is coming where, it was kind of impossible to determine which patient would come when. So I had to come every day for a lot of hours and just like test my luck if I would find a sample or not. But so yeah, this is one of the constraints we have with LMICs. And so the global prevalence of PCOS is 4 to 20%, whereas in Pakistan it is a staggering 50 to 55%. Even I have PCOS. Half of my friends have PCOS. It's extremely common here. And there is a lot of stigma around fertility, especially in a country like Pakistan. There is a lot of stigma around the diagnosis. They don't want to get diagnosis. They think it leads to infertility, which is again not the case at all. But there is the social stigma that we have around this, and also because of the higher prevalence, we are at more at risk. Pakistani women are more at risk for this. However, we don't know why. We don't know why, and we don't know what will happen to us if we have PCOS, right? So it's like a very bleak situation right now in a way that I really hope to contribute more to it. And I am a big advocate on these issues. But like we don't know if it could cause cancer or not. We did not know if it could cause endometriosis or not. We did not know if our genotype or genes that were expressed in our population were different from the Western data sets or not. So again, this is like a huge gap that I did try to fill, but there are, and I will talk about more gaps also. So again, molecular extraction, so my lab was pretty underfunded, so I had to do everything myself. We had no kits and stuff, so I did the RNA extraction through Tyrol method, cDNA synthesis, and then we ran it gel PCR, electrophoresis, etc. And then we quantified it with qPCR. I normalized it with ACTB and GAPTH. And then this is how I did my research. So again, we found out that there was, as you can see, P10, which is extremely, which is a gene that, if downregulated, can lead to cancer, was downregulated in cancer like expected, but it was not that downregulated in PCOS. There was a there was no significant difference between that. So that's kind of a good thing. I would say that for now, we do have some evidence that PCOS may not lead to reproductive cancers. However, there was this link. As you can see, there was this link between so this is the ESR1 expression, which is the estrogen receptor. And we did see again that there was a significant underexpression of ESR1 in reproductive cancers. But again, as PCOS has been determined as an endocrine disorder, we did not still see ESR1 downregulation as expected. There was still some downregulation, but not significant enough. Then we did also have NFE2, the oxidative stress response downregulation, was noticed more in PCOS than others. So that did lead to the determination of that maybe there's some implication. There were some other genes that we found out not to have significant changes in either cancer or PCOS with control. For example, we had SAW2, IGF-1, IGF-2, all of them, they were not, and by the way, they were also not showing much in the gel, and they were not quantifiable also, because I was doing it through blood, and these modulations are usually in the tissues. So I think I would have like a better research if I did get the hands-on tissues. But again, that was something that I was not able to do during my undergraduate studies. So again, this is just like some demographics of my patients. So we had this is actually 20, by the way, sorry. But so we have 20 patients, PCOS patients. We had 42 patients with reproductive cancers and 8 healthy controls. And I was actually nicknamed Mosquito in my university because I used to just get people, you want to give blood? I want to run experiments on it. It was very funny though. So so control was from my university. We also the PCOS samples they were quantified through some hospitals, and then I did have some samples from my university that I validated through their um, reports and everything. Now, we do see that there is oxidative stress both in PCOS and in cancer. We do know that there's not a big link between them. But again, the oxidative stress will lead to something. But it's not cancer, so what is it then? I mean, personally, I think it's diabetes because everybody has it here, but I'm not sure. This is something that I really want to do for my next studies for my master's thesis. So we do see this. Whereas, so again, one of the limitations that I had again was I wanted to that I've ran it on blood samples, which is good or bad if you see, because again, blood samples would of course be subject to RNA degradation and stuff like that. But it could also mean that if we are able to find a blood-based biomarker, it's easier because it is way more difficult to get a tissue sample than to get a blood sample. And if you're able to determine the problem through blood sampling, I think it's more accessible, it's more convenient, and it's easier to do it, especially in such settings where this is already something that is different from what you've known. So again, this was my research. I really hope to take it. I really hope you enjoy it. I know it's a bit different approach, but I'm open to questions.
Thank you so much, Simil. There is a question in the chat for you from Emily, and it's actually a really similar question to the one I had, so I'm very happy to see that. And if you don't mind, you can just answer maybe in the chat. I will put up my slides for the closing remarks, but for those who are interested, you can stay on for a couple minutes and chat with Simil as well.
Yeah, let me see.
Yeah.
Um,
So
should I
I
stop sharing?
just if you can stop sharing and then you can answer Emily's question in the
Yes, Simran,
chat
you have a
directly.
question?
Okay, so I just want to thank you again, all the presenters, and to everybody who joined us today, whether this was your first session or you've been part of both. I hope you took some really interesting takeaway messages because I know I did. If you'd like to keep the momentum going, we encourage you to join the Women's Health Research Cluster. Membership is free and it's an easy way to stay connected to this growing community and future opportunities. So you can just scan the QR code on the screen here to sign up. And thank you again.
Simran has a question.
Yes. Simran, if you'd like to ask,
Oh, yeah. Sorry, I can't turn my camera on right now.
all good.
I would just like to comment. Firstly, I greatly appreciate this because I think it's not a very well-known fact that South Asian women have staggeringly higher prevalence of PCOS. I myself have PCOS, and so do four different women in my family. I was wondering if so when you're looking at these specific genetics and like the downregulation and upregulation of it, were you just looking at these specific genes associated with oxidative stress, or are there other mechanisms you considered or are thinking of considering when you're looking at how PCOS and like things related to it may have a connection to gynecological cancers?
That's a really good question because yes, again, this is an issue that we from South Asian descent face a lot more than other people, and a lot of times I tell people I have a symptom like, oh, you don't have hair on your face. I'm like, that's not it. That's not a symptom, or not really that big of a symptom or something like that. It's extremely, it's I think it's something that especially women here face a lot. Secondly, yes, I did see a lot of other mechanisms also. So there were 8 genes in total that I looked at. There were two for cancer. There were two for diabetes. And I wanted to see this because I really think there's something out there, but I could not find much because it wasn't I wasn't getting good results in blood. So maybe I would have to look at tissue. And two genes were for estrogen hormone, and then two were for actually one was for estrogen and three were for oxidative stress. And there was upregulation for some and then for the oxidative stress in PCOS. Again, uh, I wanted to do again. This was a thesis, and I already went pretty overboard with what I was doing. Uh, I don't know how my supervisor allowed it. I think he was just aloof about it until I did the work. I was just like, I'm doing it, I'm doing it. But yeah, so this was something that I had limitations in that. I really want to see its association with diabetes. I really want to have a cohort of diabetes, and I want to see what genetic patterns that they have and similar, because I really do think that there's something out there for that. I know there has been some talk about the risk factors with heart. I'm already replying to it, but I think heart factors will have an effect also, but from what I think, because insulin resistance is a common factor in both diabetes and PCOS, and from what I've noticed, I've started to do a bit of research on this, is that people with PCOS do develop diabetes earlier than expected. Usually, people with South Asian descent will eventually develop diabetes, but they don't do it until they're 50, in their mid 50s or 60s. But with PCOS, I've noticed that one, they definitely 100% of the times do develop gestational diabetes. Secondly, they do end up getting diabetes way earlier. So there's something out there for that. I really hope to see what it would be, but yeah, I hope that answers your question.
Yeah, thank you very much. Definitely also very impressive work for undergrad. Um,
I don't know how to do it.
uh, yeah, and I definitely do agree with the aspect of insulin resistance, as that's like a shared through line between not just diabetes, but also uh, premature like female obesity, and like the body weight aspect of it and the fat repository aspect of it is a very common thing that's shared between the two. And I know it's definitely difficult to get significant sample sizes in South Asian cohorts, because uh, that's not something that really is prevalent right now, even in studies that do look at this. So I definitely, I really appreciate it. I'm excited to see what else you have coming up.
Thank you so much. Again, like I said, the main problem that I had was that every data was very manual. I had to whisk through it. There were like 40 pages for one patient, and to select what to look for, what not to. And I think it's a good thing also, because I have a lot of data now. But I had to whisk through it, and everything was extremely difficult for me to do, because they were like a store full of files that I had to whisk through. And it was not good for my dust allergy also. But I think that although there is, so this just means that this is more opportunity for me. And I just hope that everybody gets together for this, because this is something that I don't think should be ignored for longer. But thank you so much for your question.
All right, thank you so much. So with that, we'll just close out for today's event. And yeah, thank you again for joining.